Welcome back to the Field Design and Analysis Workshop. So now what we're going to do is use a different data set, estimate some summary statistics, and also perform an ANOVA using R. Okay? So the data that we're going to use came out of a project that was funded by USAID through a USDA project, and I'll give some more credit as we go. So we're concentrating on this box in the upper left-hand corner. Okay, so particularly we're going to be looking at distributions and analysis of variance, and then partitioning variation and using that to estimate heritability, as well as estimating best linear unbiased predictors for our traits. All right. So at the end of this module, you're going to have a better understanding of how to structure data for statistical analysis. And for this point, it doesn't matter if you're using R, SAS, or another program. Okay? It's the structure of the data so that it's analysis ready. We're going to read data into R. We're going to do some rearrangement of that data and checking it using R. Okay, and again, this is an important feature of R, is it allows us to do some manipulations of the data. We're going to add a package, maybe two, to do some analysis. And then we're going to use R to visualize the distribution of data. We're going to understand how we can transform data using R, and then perform an analysis of variance using linear regression with fixed effects in the R core package. Okay, that's where we want to go. Okay, so we will be talking about both fixed effects models and random effects models. All right, I use this chart to kind of help me figure out what we want to do. So if I hope to consider that my data is a sample from a probability distribution, chances are I'm down this random track. Okay? However, if I really want to know whether two of my treatments are different from each other and it's only those treatments I care about, I'm probably going to be on a fixed effect track. Okay? So some general guidelines for helping us to make some decisions. So if we're going down the random effect track, we're interested in the distribution of those random effects. In other words, estimating variance. Okay? If we want to calculate the predictions of the realized values, these would be BLUPs. Okay? Then we're on the random track. For this example, we're going to be using data that comes from a group at Michigan State University. So it's a group led by Jim Kelly, who's the breeder, and then Martin Chilvers is a plant pathologist who works with him to design germplasm screens. Jacobs, Byrne, and Wright are staff that work with Jim and Martin. Okay? And so what we're going to be dealing with is a set of data that comes from screening Andean dry bean germplasm for root rot resistance. In the context of this project, Martin conducted a survey in Michigan, Rwanda, and Uganda, collecting isolates that were causing root diseases in the field. And so what he was seeing was a lot of Rhizoctonia, Pythiums, and Fusariums. Okay, so 2 true fungi and then 1 oomycete. All right? So Rhizoctonia turned out to be the most common cause of diseases, but even though it's a single species, there were multiple anastomosis groups that were showing up. So a single species accounting for most of the root rots, but diversity within that species. Okay? Fusarium, 9 different species. And then if we look at the oomycetes, so 9 Pythium and 2 Phytophthora species. Okay? So because of the species diversity in this group and also the predominance of Rhizoctonia, even with the variation of anastomosis groups, Rhizoctonia seemed like the more tractable organism to go after first. Most common, least amount of species diversity. Okay, so in terms of screening plant genetic resources or germplasm for resistance, we need to set up an experiment that's designed to separate variety or accession effects from environmental effects. So these variety or accession differences are genetic differences. These are our treatments. Okay? So in order to do that, it requires that we have a design that controls for treatment and environment. Right? Okay. So if we want to treat this as a fixed effect, we're really asking the question, are any of the accessions resistant? And maybe even which accessions are better than others? Okay, so this is a perfect fixed effects question to ask. If, on the other hand, we want to know what the proportion of variance due to genetics and environment is, we're in that random effect question. Okay, so most statisticians are going to tell you that you should know going into the experiment whether you're working with fixed effects or random effects. I tend to look at this from a tool user's point of view. I can get different information out of the experimental design by running it as a random effect than if I run it as a fixed effect. Okay? So I'm approaching this from the point of view of what information can I extract from the experiment.